Our next unit is on amino acids, peptides, and proteins. Proteins are biopolymers of alpha amino acids. Okay, that means that there's an amino group on carbon number 2 of a carboxylic acid. Amino acids are joined to each other to form a peptide and a protein by what are called peptide bonds. The peptide group is actually an amide group. Some of the functions of proteins include structure. Okay, this is keratin and collagen, hair, skin, nails, for example. Enzymes, these are extremely efficient catalysts in the body. Transport, this would be like hemoglobin. Protection, antibodies, primarily what we're talking about here. Hormones, and under protection, possibly venoms and toxins. Okay, so here we have a general alpha amino acid. Consists of 4 parts: the carboxylic acid group, the amino group, and the side chain, and the usually a hydrogen. Okay, with one exception. Okay, so here is the amino acid alanine, and these are named based on what the side chain is. So you have alanine, serine, glycine, cysteine, and valine. And if we split out water and we reconnect these, we get what are called peptide bonds. And the peptide bond is actually an amide group. Okay, amino acids have an NH2 group on the carbon next to the carboxylic acid group. And these are all primary amino acid groups except one, and that's proline. Glycine, where the side chain is a hydrogen, is the simplest. And this is the only one that doesn't have a chiral center. Okay, except for glycine, with R as a side chain, the molecule will be chiral. Most natural amino acids are L amino acids. That is, they're related to L minus glyceraldehyde. The direction of optical rotation, plus or minus, again, must be determined experimentally, and we cannot predict that from D or L. Okay, so here we have L-alanine, and the L indicates that when we draw the Fisher projection, okay, we place the carboxylic acid group at the top. In the Fisher projection, the amino group would be on the left. Okay, this is related to L-glyceraldehyde. Okay, there are 20 standard or common alpha amino acids, and they differ in side chain characteristics. Some of them are nonpolar. They have a hydrogen or an alkyl group. Okay, some of them contain OH. There are some that contain sulfur. There's one that's a thiol, and then there's one that's a sulfide or thioether. Some of them contain a non-basic nitrogen. Some of them have a carboxylic acid group on the side chain, and some have a basic nitrogen. Okay, of these 20 common amino acids, especially in human protein. 10 of those are what we call essential amino acids. The name essential can be somewhat misleading. The non-essential amino acids, which would be the other 10, do not indicate that we don't need them. What that means is that for the essential amino acids, these have to be taken in through the diet. In other words, our body cannot make these. Okay, the non-essential amino acids can be made by the body. Okay, so here we have a table of the various amino acids, and these are grouped according to whether the side chain is nonpolar, whether it has an OH. We'll look at sulfur-containing side chains, carboxylic acid, and nitrogen-containing side chains. Okay, the various columns. Okay, there's two types of abbreviations. Let's go back. There's the one-letter and the three-letter abbreviations. The one-letter abbreviations are becoming more common. Okay, and the side chains are identified here. This is the only amino acid that has a secondary amine. It's proline. It's got a rigid cyclic structure. And then this is what we call the isoelectric point, or the isoelectric pH. And what that means is that this is the pH at which the charges are balanced. Okay, so we have an acidic group and we have a basic group, and the basic group can pick up a proton from the acidic group, and at this pH, that's the dominant species, where we have a protonated nitrogen and an ionized carboxylate. Okay, here are a couple of amino acids that have alcohols on their side chains, serine and threonine. Okay, and here's a phenol, tyrosine. And we've got a couple of these that contain sulfur. Cysteine has a thiol. Methionine has a thioether or sulfide group. And then we have some that have a non-basic nitrogen. Okay, this is a non-basic nitrogen because it is an amide. So is glutamine. And then tryptophan has a non-basic nitrogen because protonating this nitrogen would destroy the aromaticity of this ring. Okay, and you'll notice that most of these isoelectric pHs so far are in the range of about 5 to 6. Okay, and then we have the acidic side chains, aspartic acid and glutamic acid. And notice here the isoelectric point drops considerably. And then we have some amino acids that have basic side chains. And here you can see that the isoelectric point goes way up. It's a higher pH. Right, a complete protein is one that has all of the essential amino acids. And examples of complete proteins are those found in meat, fish, milk, and eggs. Plant proteins are generally incomplete. Okay, that's not always the case, but in general. And then vegetarians should eat many different kinds of plants. For example, beans and rice together provide a complete protein, but neither beans nor rice by themselves are complete. Okay, these are some examples of rare amino acids. We have 4-hydroxyproline and 5-hydroxylysine, which are found in collagen. D-glutamic acid is found in the cell walls of bacteria. Okay, most common amino acids are L. D-serine is found in earthworms. Gamma-aminobutyric acid is a neurotransmitter. Beta-alanine, okay, where the amino group is on the third carbon rather than the second, is a constituent of vitamin pantothenic acid. Okay, when I said serine just a moment ago, my iPhone went off. Okay, amino acids have high melting points, usually over 200 degrees. Excuse me. They're more soluble in water than in ether. They have larger dipole moments than simple acids or simple amines. They're less acidic than most carboxylic acids, but less basic than most amines. And okay, at the isoelectric point, this is the dominant species where both of these groups are charged. Okay, this is called a dipolar ion or Zwitter ion. Okay, so at the isoelectric point, the dipolar ion is the dominant species. In strongly acidic solution, this will be protonated and this will be protonated. In strongly basic solution, this will be neutral and this will be negative. So amino acids exist as these dipolar ions. The carboxylic acid group is acidic, it loses a proton, whereas the amino group is basic and gains a proton. The actual structure, whether we have ionized groups or not, will depend on the pH. Okay, so here is a titration curve for glycine. And under strongly acidic conditions, okay, both the carboxylic acid group and the amino group are protonated. Then we go through a buffer region, and at the midpoint of this buffer region, pH equals pKa, so that's 2.3. That's the pKa for this carboxylic acid. Okay, and then we reach the isoelectric point where this is the dominant species. Okay, and then continuing the titration, we go through a second buffer region. Here, the midpoint of this buffer region, pH equals pKa2. And then above that, this will be anionic, above 9.6. Okay, at the isoelectric point, or pi, this is defined at the pH as the pH at which the amino acids exist as the zwitterion. The charges are balanced, so overall the zwitterion has a neutral charge. And the actual isoelectric point depends on the structure of the side chain. Okay, for acidic amino acids, those with an acidic side chain, the isoelectric point is about 3. For basics, it's about 9. And for most of the others, it's about 5 to 6. Amino acids can be separated by electrophoresis. And this simple diagram shows that if we use a pH 6 buffer solution and we want to separate 3 amino acids, okay, for alanine, we're at the isoelectric point, so that doesn't move. Okay, aspartate moves toward the positive charge since it has a net negative charge, and lysine moves toward the negative charge since it has a net positive charge. Okay, opposite charges attract. Okay, in the next section, we'll look at some reactions of amino acids and methods of preparation of amino acids.